Hello viewers, today I have come with my new video with a very interesting topic, that is contraction of skeletal muscle. That means how our muscle contract. When we walk, run and do some physical workout, our skeletal muscle need to contract to promote that work. So we must know the physics and chemistry behind this biological activity. So let's start. Before getting into the mechanism of skeletal muscle contraction, I want to clear you the structure of skeletal muscle first. So here it is. Here in this picture we can see that this is the bone and this is the muscle. Okay. So this muscle is attached with the bones by means of this structure. This is called the tendon. It is actually the collagen, made up of collagen. Then tendon, within the tendon there is a muscle membrane that covers the inner part of the muscle. This outer membrane is called the epimysium, and within this is a bunch of the fascicles that surrounds by a membrane called the perimysium, and perimysium holds these bunches. This bunch of myofibrils, this is called the fascicle. This is called the fascicle, or we can say muscle bundle. And within this muscle bundle there is a endomysium that surrounds the muscle fiber. So these are the muscle fiber. Muscle fiber is surrounded by a membrane called sarcolemma, and the cytoplasm behind this is called the sarcoplasm. And within this muscle fiber there are myofibrils. We all call them as a myofilament, and myofilaments are of two types. These are there is striations, actually dark and light bands are there. Thick filaments are called myosin, and thin filaments are called the actin. Cytoplasm of the skeletal muscle, that is the sarcoplasm, it contains the mitochondria and more nucleus. Okay, and the mitochondria of the skeletal muscle is called the sarcosome. Syncytial structure is actually we can find in the skeletal muscle. Syncytial structure, that is syncytium. What is this? This is actually when during cell division, if there is no cytokinesis, that means cytoplasm, if cytoplasm not divide, then only nucleus divide to form more nucleus within the cell. So a cell contains more nuclei. If there will be cytokinesis, then the cell will be separated within a single nucleus, but taking single nucleus inside it. But if the cytokinesis not occur, then a cell contains more nuclei. That condition is called the syncytium. And we can see in the muscle cells this type of structure, syncytial structure. That means muscle cells are muscle cells with numerous nucleus within sarcoplasm. Another more important structure of the skeletal muscle is the presence of sarcoplasmic reticulum. This is, if we cross section the skeletal muscle, we can find this type of diagram. That is this is the sarcolemma, the membrane of the skeletal membrane of the muscle fiber. And there is an invagination. This invagination or that means the sarcolemma come inside the muscle fiber and form a invagination. This is called the T-tubule. And this is the sarcoplasmic reticulum that forms a network like structure beneath the sarcolemma. That the function of the sarcoplasmic reticulum is mainly to store the calcium ion, which is this sarcoplasmic reticulum is actually it's a modified form of endoplasmic reticulum. Okay, it helps it actually supplies calcium ion during contraction. And beneath this sarcoplasmic reticulum, there is a parallel bunch of the myofibrils, alternative dark and light bands are there, and these are the uh, dark bands of adjacent myofibrils. They are actually lie side by side. Here we can see that dark bands are lying side by side and light bands are also lying side by side. That's why they are forming a striation. So forming the striations or strips. That's why these muscles are called the striated muscles, because of having these striations, dark and light bands. Here is another structure that is called a dystrophin protein that also help in contraction. It relay the signals to the endoplasm reticulum for releasing calcium, and also whenever there is much more calcium, it also relay the signal to the endoplasmic reticulum. So these are the dystrophin protein located here. Now we come in detail with single myofibril. That is a single myofibril. Here it is the sarcomere. Now we are coming to the details of sarcomere. Here is the structure of sarcomere. So these striations, dark and light band, the half of the dark band from one dark band and another for the next dark band, half of the next dark band. This portion is called the sarcomere. This line that cross the between the dark band is called the Z line. So the distance between two Z line is the sarcomere, which is actually the functional and structural unit. So this sarcomere, the dark and light bands, it's made up of two types of protein. These are the thin filament, that is light bands. Here I draw is as a blue color, in blue color, and this is the thick filament. This thin filament is made up of actin protein, and this thick filament which is made up of myosin protein. Okay, now look at here, the middle line. This middle line is called the M line, that pass through the middle portion of the thick filament. This portion between the gap between these two actin thin filament is called the age zone. This sarcomere is actually, it's a whenever the muscle fiber in resting stage, the muscle fiber looks like this. This is the A band. A band contains the thin and thick both of the filament. Look at this picture here. If we this is the A band, so this portion is the A band. Within this part there is a light band, also the dark band. The light band, also dark band, light band, dark band, light band. So A band contains both the filament, dark and light. I band is this portion. Here only thin filaments are there. Only thin filaments, this portion. This is the I band. Only these portions here. Okay. And Z line is the line that crosses at the half of the thin filament. And M line is passing through the middle portion of the thick filament. So these are different bands and lines, Z line that bisect the thin filament. Okay. So these are the different bands. Z line is also called the Krause's membrane or Dobbe's line. Okay, Z comes from the German word Zwischenschläge. That means between deals. And M, the word M comes from the German word Mittelstäbe, that is central disc. So these are all about the sarcomere. Now I am coming in detail with these two filaments, that is thick filament and thin filaments, because these thick and thin filaments are made up of proteins. And these proteins, actin and myosin, these proteins are called the contractile proteins. So let's come. Here is the actin fiber, the structure of actin fiber. It is made up of two polypeptide chain which intercoiled with each other. and form intercoiled with each other and form a double helix. This is a one polypeptide chain, and this one is another polypeptide chain. So these are two polypeptide chain that intercoiled with each other. Actin fiber has three part. Actin fiber has three part, that is three types of component: actin, tropomyosin, that violet colored. These tropomyosin is also made up of two polypeptide chains that uh, covers the active binding site of the actin, and troponin. This is the troponin. This is also made up of three components. Okay, I am coming in details. So first I am telling about the actin. The unit of the actin, this polypeptide chain, single unit monomer, which is called the G-actin. G-actin by polymerization, that means more than G-actin together form this polypeptide chain, or this is also called the F-actin. So G-actin is actually the polymerized form of the F-actin. That means G-actin polymerized to form the F-actin. Here G-actin has a site that binds with the ADP during muscle contraction. Next, tropomyosin. It is formed of two polypeptide chain, I have already told you, and it's a spirally coiled around the F-actin. This is this violet color. This spirally coiled over the around the F-actin. That means this polypeptide chain, and it lies in the group of the F-actin in such a way that each tropomyosin molecules covers 7 active sites of actin molecules during relaxed state. The 7 active sites are here. These are covered by this tropomyosin in relaxed state. Next come to the troponin. It is a globular protein and lying at the end of the tropomyosin protein. Troponin has three units, I, C and T. Tropomyosin binding troponin, that is TPT. Calcium binding troponin, that is TPC, and troponin inhibitor, that is TPI, which blocks the myosin binding active site of actin. So these are all about the actin fibers. Now come to the structure of myosin fiber. This is the thick filament. Myosin protein is a type of ATP dependent motor protein. It about 1500 Armstrong long and 20 to 40 Armstrong in diameter. Okay, it is a asymmetric molecule. And is formed of 6 polypeptide chains differentiated into two parts. That is, this is the tail. This part is the tail, and this is the head. Tail and head. Tail is formed of two polypeptide chains, is a monomeric protein called meromyosin, intercoiled upon each other to form a double helix and form light meromyosin. And heads are 2 in number, and it's also made up of two globular meromyosin proteins, which forms the heavy meromyosin. Heads lie at the tip of the small sized arm, and it's form the cross arms. It forms cross bridge with actin filaments in presence of myosin ATPase present on the heads. Here is two part on their head. That one part is actin binding site and another part is ATP binding site. During contraction, this head attach with the actin filament by this site, and ATP binding site is here. Here ATP binds with the myosin. Okay, So the myosin proteins are responsible for contraction and relaxation of the muscle. So these are all about the structures of the skeletal muscle and their fibers containing proteins. So now I am coming in details with the mechanism of this mechanism of contraction of the skeletal muscle. So most satisfactory theory of muscle contraction is sliding filament theory. Okay, proposed by A.F. Huxley and A.H.E. Huxley and J. Jansen. It is based on the structural changes in binding pattern of the sarcomeres of the myofibrils. Of the striated muscle fibers. Okay, so it actually depends on the basis of some changes. They actually propose this theory based on some statement. The statements are: length of a band remains unchanged. During muscular contraction, the length remain unchanged. The length of A band remains unchanged. This A band, this length remain unchanged. Distance between the adjacent edge spaces remain constant. So this edge space, that space remain constant, and the line M line of the adjacent, because there is another muscle fiber, another M line. So this distance remain constant, and the edge zone, the distance between the edge zone, it decreases, and the distance between adjacent Z line, this is a Z line, and this is another. Between these Z lines, that also size of this distance between these adjacent Z lines and the size of the sarcomere is decreases. So based on this statement, on these changes, they propose the sliding filament theory. This decrease the length of the sarcomere without any change in the size of A and I bands. A similar action is in all the sarcomere results in the shortening of the whole myofibril and also thereby the whole muscle fiber. So before contraction, muscle contraction, signal must come to the muscle, otherwise muscle can't be contracted. So here is a neuromuscular junction from where the signals comes. So this is the motor nerve and this is the skeletal muscle, and the junction between this nerve and muscle is called the neuromuscular junction. The stimulus comes from this motor nerve. When the motor nerve gives signal, the neuromuscular, okay, I'm first explaining you the neuromuscular junction. So here is the neuromuscular junction. This is the motor nerves. Okay, motor nerves, this is the action terminal, and this is the myelin sheath. Within the action terminal there is a synaptic vesicle, and within the synaptic vesicles there are neurotransmitters, and this is called the presynaptic membrane, and this is the postsynaptic membrane, and the gap between these two is called the synaptic cleft. This is the muscle membrane of the muscle. Okay, and these are some mitochondria, because we know that skeletal muscles contains more mitochondria because it needs more energy. So this is the mitochondria, and this is the subneural cleft. This is the synaptic cleft and this is subneural cleft. Okay, so here it is these are the synaptic vesicles. Whenever the signal comes to the nerve, the synaptic vesicles come in the presynaptic membrane and it releases the neurotransmitters. And these neurotransmitters contains acetylcholine. That acetylcholine comes in contact with the muscle, and muscle also have some receptor that receive the acetylcholine. So there are acetylcholine receptor on the T tubule. I have explained already that there is a invagination on the sarcolemma that form the T tubule. Within the T tubule there is a acetylcholine receptor. That acetylcholine receptor receives the acetylcholine. Okay. So here the stimulus comes and the contraction procedure generated. When the when this acetylcholine binds with the receptor of the sarcolemma become more permeable to sodium that actually calcium ion due to opening the sodium channel. Sodium channels get opened and the sodium get entered into the muscle, and sodium rapidly diffuses into the concentration gradient, and electrical gradient is formed, and this is actually form the depolarization of the sarcolemma. Actually, in resting stage muscle fibers, sarcolemma is electropositive on outside and electronegative from the inside. But when this acetylcholine come and combined with the receptor, that signal stimulate to open the sodium channel, and sodium get entered into the cell. So whenever the sodium get entered into the cell, it forms the inner side. It forms the inner side positive and electronegative at the outside. Such sarcolemma is called the depolarized. Okay, and a new different potentiality is actually formed, which is called the action potential, and this is due to the signal. And this action potential which is formed here, which is go through the length of the myofibril, and that cause the muscle fiber through the length of the muscle fiber by the T-tubule system, and that stimulates the whole muscle fiber. And whenever this action potential generated, this sarcoplasmic reticulum, which stores more calcium ions, that release calcium ions. Okay, that sarcoplasmic reticulum release calcium ions, and they initiate the biochemical changes in the muscle contraction. Because this calcium ion, after getting released from this part, it will go to the to combine with the myosin membrane. So by stimulation, the calcium ion get released from this sarcoplasmic reticulum, which is stored in the sarcoplasmic reticulum. Okay, when calcium releases from the sarcoplasmic reticulum, it comes and combines with the troponin, where the calcium binding site is there. Okay, here I am showing you the muscle before contraction and during contraction. Before contraction, this is the myosin filament, this is the head, and that is the tail. Okay, and this is the actin, this is the tropomyosin, and this is troponin. Myosin head, troponin. So this is the condition before muscle contraction, normally original state. And here is during muscle contraction, what happened? 4 calcium ion comes and combined with the troponin. Look at here in this picture, there is a site of calcium receiving site, receptor. C means calcium binding site. Okay, so here calcium binding site is here, and calcium comes and combined with this receptor. And whenever this calcium combined with this receptor, it replaces from this original position, and it opens the head of the actin binding site. I repeat, when 4 calcium ions combine with the troponin C, then the latter, it moves the tropomyosin molecules deeper into the groove between 2 actin strands. This actually uncovers the active site of actin molecule to permit the binding of meromyosin head to myosin protein. Now, ATP hydrolyzes to form ADP and inorganic phosphate and release energy, and this energy stimulates the formation of actin myosin complexes between the heads of myosin, active site on actin filament. Due to interlinking, the head are tilted. Now I am showing you this function, this activity on my next diagram. So this is the normal stage. This is the normal stage, the tropomyosin, the head of the myosin. Now the ATP comes and release energy. By getting this energy, the head of the myosin get attached with the binding site. First their position is here. Now they get energy, and when the calcium comes in contact with this site, troponin, that also uncovers this myosin filament, and then whenever it uncovers the actin, then the myosin head get attached with the binding site. Here, look at here, the myosin head is attached with the actin. Okay. Whenever they attached with the actin, they forms a actomyosin complex. When the head get attached to the they form the actomyosin complex. Due to this interlinking, the heads are tilted, and this tilt is called the power stroke. It occurs at the release of phosphate. Okay, so the myosins function as the diving motor of the filament sliding. ATP detaches during the pivoting of the myosin head. When the myosin get attached, and they tilt the actin fibers here towards the M line, and then ADP detached. Next, another molecule, whenever the another ATP hydrolysis, it release more enzyme, and that more energy, and that energy, by using that energy, the myosin head tilted more, and the actin filaments slide inward more and more. And that rotation, myosin, I am repeating that whenever the another ATP molecule hydrolysis, that release energy, and that energy, by using that energy, myosin head repeat. Myosin head rotate, and it slide the actin filament inward. That means towards the M line. Look at the position here. This is the myosin fiber, and this is the actin fiber. Whenever the power stroke occur, and myosin filament slide the actin filament from this side to this position. So the actin filament come towards the M line. This is due to power stroke. This is the slide. Myosin filament slide the actin filament towards the M line, towards the inner side. Okay. And this combined effect of power strokes causes muscle to contract. And this cause the shortening of the myofibrils called contraction. The shortening of all the myofibrils decreases the whole muscle. Look at this picture. This is the normal sarcomere. This is Z line. The lengths are normal for all bands. Now this is the contracted sarcomere. The gap, the H zone get decreased, length decreased. The length of the A band remains same. Okay. The I band, length of the I band remains same. And the actin and myosin filament get attached to form the cross bridge. This is called the cross bridge. Whenever the head of the myosin filament get attached with the actin, they form the cross bridge. By forming the cross bridge, the actin filament get tilted and get slide. Okay. So after maximum muscle contraction, calcium from the sarcoplasm is trapped by the sarcoplasmic reticulum by active transport, which actually inactivate the myosin ATPase enzyme. So no energy is available, and the actomyosin complexes dissociate, and by this dissociation, then the original position of the muscle comes. Here we can see the dissociation. The actin get released from the myosin head. When the calcium get back to the sarcoplasmic reticulum, so the troponin get free. It come to its original position. Then it's again covers the actin binding site, myosin binding site of the actin. And whenever this tropomyosin covers the actin filament, then the myosin head get detached from the actin filament. That detach is called the dissociation. So muscle fiber have the ability to excite and contract, and that response comes from the motor nerve. And that duration up to which the muscle can contract, up to which how long the muscle can excite, that actually depends on the supply of the ATP. If there is much more ATP, much more energy, then the actomyosin complex will form and the contraction will be continued. But if there is a limited amount of energy, then there is no contraction, no excitability we can find in the muscle. So when ATP is there, the muscle will contract. One more thing here I have to mention, that if here in the contraction site, generally 4 calcium requires to combine with the troponin, but if the concentration of the calcium ion become more, that also gives a signal by the dystrophin protein to the sarcoplasmic reticulum that there is a more calcium ions. You have to stop releasing calcium ion. Then the calcium ion gets stopped releasing from the sarcoplasmic reticulum, and the steps are occur accordingly. Whenever the calcium stop releasing, when the sarcoplasmic reticulum stops releasing calcium ions, then there will be no calcium ion binding with the troponin. If there is no troponin binding site, calcium binds with the troponin, it will not uncover the actin filament. So the myosin filament will not attach with the actin filament. So this process is happen, the dissociation. That means that the dissociation is actually called the relaxation. The muscle will relax. So for contraction we need calcium ion, and for relaxation we don't need any calcium ions. So here one more thing is I have to mention that if we do heavy exercise, our muscle get fatigued. Why it's happen? Because when we do heavy exercise, we do heavy workout, lactic acid actually formed in our muscles, and that lactic acid formation is faster than its oxidation. So muscle come in oxygen depth condition. Muscle fibers undergo oxygen depth condition, and it decreases the ATP supply to the muscle fiber, and it will not promote the formation of actomyosin complexes due to the absence of ATP. So the muscle will not contract, it will become fatigued. So in this way muscle contract. I have explained you in detail. Go through my diagrams and also my audio. It will help you very much. If my video help you, then please like, share and subscribe. I will come very soon with my new video. Thank you for watching. Take care.